Okay, so in this video we will see complete details about gel electrophoresis, how gel electrophoresis run, and what are the requirement to run gel electrophoresis. So let's start the video. So here you can see a setup of gel electrophoresis. This is a matrix. Okay, these two are electrodes. These electrodes, this is negative electrode and this is positive electrode. Okay, these are attached to this gel tank. Then this is a matrix where DNA will run. In this matrix some wells are created. Okay, some wells are created. In this well DNA is added. Okay, then after supplying the current, DNA will move in this way towards the positive direction. Why? Because DNA are negatively charged due to the presence of phosphate in their backbone. So there will be repulsion between negative and negative charge, and DNA will move in this direction. Okay, this matrix can be made up of agarose, or polyacrylamide. Okay, polyacrylamide. This can be made up of agarose and polyacrylamide. Okay, so let's see when the matrix is made up of agarose or when it is made up of polyacrylamide and how the gel electrophoresis works. So here you see gel electrophoresis. It is used to separate a mixed population of macromolecules such as DNA or proteins in matrix. Okay, suppose there is a mixture of DNA. Okay, there is a mixture of DNA. Some are larger DNA, some are small DNA, and we want to separate this DNA from this mixture of DNA. Then we will run gel electrophoresis. And then the heavier DNA segment cannot move a long distance, and small DNA segment will move to a long distance, and in this way we can separate our desired DNA by its mass by running gel electrophoresis. We will see this in detail. Okay, separation is done by applying electric field. Biomolecules are separated by size. Okay, so let's see this in whiteboard. Okay, suppose this is a matrix. Okay, here one DNA strand and this is another strand, this is another strand. Okay, so obviously heavier particles cannot move for a long distance. Okay, and the light molecules can move a long distance, so it will move up to here, suppose, and this will move here, and this is again small, so it will move like this. Small or low weight molecules or DNA segment move longer. As much as it is small, it will move longer, and a heavier particle cannot travel a long distance. Okay, this is heavier particle, so it cannot move a long distance. This is smaller, so this moves long. Okay, then migration is expressed in terms of electrophoretic mobility. Remember this formula: V by E is equal to Q by F. V is rate of migration, E is strength of electric field, and Q is charge, F is frictional coefficient. Okay, when DNA, suppose this DNA is moving from here, it is moving. Okay, then rate of migration, at what speed it is moving from here to here, it is denoted by V. That is rate of migration. Okay, at what speed DNA is moving. Then this particular DNA will also have some negative charge. Okay, that charge is represented by Q. Okay, after applying the electricity, it will create an electric field here. Okay, it will create an electric field here, and the strength of the electric field is denoted by E. and when DNA is moved from here to here, an opposite frictional force will act, on this DNA. Okay, that frictional force or the coefficient of that frictional force is represented by F. Okay, frictional force coefficient. Then there are two types of gel electrophoresis. One is agarose, one is polyacrylamide gel electrophoresis. So agarose gel have larger pore, separate large DNA, large molecules such as DNA, and composed of agarobiose, okay, galactose and 3,6 anhydrogalactose. Okay. So agarose gel, when we need to separate DNA, then we will use agarose gel. Suppose here we are separating DNA. Okay, here we are separating DNA. Then this matrix will be made up of agarose. Okay, if we want to separate DNA, then the matrix must be made up of agarose. Okay, because it has larger pore size. Larger pore size means it can separate DNA. And if we increase the concentration of agarose, then the pore size of this matrix will decrease and it will inhibit the movement of larger DNA. Let's see this in whiteboard. So, okay. Suppose this is a agarose gel. Okay, wells are created here. Okay, agarose gel. If we see the agarose gel from this position, we will see the agarose gel like this way. Okay, agarose gel like this way. There wells are created. Okay, wells are created there. Okay, so it is agarose gel. Agarose is added. And when we increase the concentration of agarose, it will look like this. Okay, concentration of agarose has increased here. Okay, see, as concentration of agarose is low, here, you can see the pore size is more. So large DNA can pass through it. This. Here, when agarose concentration is increased, the pore size is decreased. So large DNA molecule cannot pass through this agarose. Okay. So depending upon the experiment, the concentration of agarose is changing. Okay. In some experiment, it may be 1%. In some experiment, it may be 0.8%. And in some experiment, it may be 2% agarose. Okay. It will depend upon our experiment, because if we increase the concentration, the pore size will decrease. That will inhibit larger molecule to pass. Okay. Polylateral crossing by B.Sc and then methylene bisacrylamide, and polymerization is started by ammonium persulfate and catalyzed by TMAT. Okay, TMAT is a chemical. Okay, that means polyacrylamide, it consists of acrylamide monomer. Suppose these are acrylamide monomer. Okay, acrylamide monomer. Then their polymerization means A will be added to acrylamide. This polymerization is started by ammonium persulfate. Okay, started by ammonium persulfate, and catalyzation means rapidly adding. This catalyzation of polymerization is done by TMAT. Okay, so here another, okay, acrylamide. So polymerization started by this and catalyzed by TMAT. Okay, then cross linking. This one strand of acrylamide and one strand of acrylamide is cross-linked by this. Okay, cross-linked by this. Cross-linked by this. Okay. Then procedure. So preparation of gel by dissolving agarose powder in buffer such as TAE or TBE. Then ethidium bromide is added to visualize DNA under UV light. Okay. So ethidium bromide gets intercalated. Ethidium bromides get intercalated into the major groove of DNA. Ethidium bromides get intercalated into the major groove of DNA. So it will help us to visualize the DNA in the final result. Okay, because finally we will see the DNA in UV light. Okay, then UV light will accelerate the electrons of ETBR to a higher position, to a higher orbit, and when the electron will return back to lower orbit, it will emit light. That we can see. That's why ETBR is added during ETBR is added to see the DNA. Okay. Okay, ethidium bromide is added to visualize DNA under UV light. Okay, because UV light will accelerate the electron of ETBR to a higher orbit, and when that electron will return to its original orbit or lower orbit, it will emit light, and we can see that. After pouring the gel into cast, a comb is placed to create well for loading of sample. Okay, so here you see, this is agarose is melting, and then agarose is poured here. Okay, agarose is poured here. Here you see, these are wells. Okay, these are wells. Here we will load our sample. And how these are created? These are created by inserting a comb here. Okay, a comb is there. So we insert that comb. Okay, there is a comb like this. We insert that comb into the gel, and that comb creates this well. Okay, for sample loading. We insert the comb into the gel like this, and that will create this well. Okay, once as the concentration of agarose increases, the pore size decreases. We have already discussed this point. And loading of sample. Once the gel has set, the comb is removed. We inserted the comb into the gel to create the well, and when the gel solidifies, the comb is removed. Okay, the comb is removed and well is created. Okay, then loading buffer is mixed with DNA, means the sample DNA containing glycerol, sucrose or ficol. Okay, why we use this glycerol, sucrose or ficol? Because these provide density. These are heavier substance. So when we add glycerol with DNA, it inhibits the DNA to come out of the well. Okay, otherwise DNA will come out of the well due to density. Okay, when we add glycerol or sucrose, it is heavier, so it goes down into the well, because glycerol or sucrose are heavier and it goes down into the well, so DNA will not come out of the well. Okay, and color dyes such as bromophenol blue to monitor the progress of the electrophoresis, whether electrophoresis is occurring or not, or whether there is chances that the machine defaults. Okay, machine may be faulty. So to know that whether electrophoresis is progressing or it is occurring or not, we add a dye, that is bromophenol blue to monitor the progress of electrophoresis. Okay, then running electrophoresis, we prepared a gel, we poured a gel, then we added the comb, and when the gel solidified, we removed the comb to create well, and after that we loaded the sample, DNA sample, along with glycerol, sucrose or ficol, so that the DNA does not come out of the well, along with some color dyes such as bromophenol blue to monitor the progress of the electrophoresis. After that, we run the electrophoresis. That means we supply current to the electrophoretic apparatus. Then electrodes are connected to that ends and voltage is supplied high voltage. Here one important thing to mind is that high voltage may hit the gel and may cause melting. If we apply high voltage, then it may cause melting. Okay, it may also reduce resolution. Okay, suppose someone is there, he wants to complete his electrophoresis quickly and applied high voltage, then what may happen? The machine may heat up and the gel will melt. Also the resolution will not be there, very poor resolution, because your gel is melted na. And if someone wants to get a high resolution and he applied very low voltage, that may also broaden the small DNA fragment. Okay, that may also broaden the small DNA fragment. Then staining and visualization. Stained ETBR is visualized by UV light. Okay, so ETBR is added, gets intercalated into the major group. Okay, major group. When UV light falls on ETBR, some of the electron of ETBR moves to higher orbit. Okay, moves to higher orbit, and when this electron comes to their original state, some lights are emitted, and this light can be seen. Okay, so this was your gel electrophoresis. Let's see the whole process in a single go. Okay, first, depending on the molecules, biomolecules to separate, we will make a gel either of agarose or polyacrylamide. Okay, if we need to separate DNA, then we will use agarose gel, and if we need to separate protein, then we will use polyacrylamide. Okay, then after preparing the gel, we will pour this gel into gel cast. Okay, we will pour this into gel cast. Okay, then we will add a comb here. And when this gel solidifies, then when solidify, remove the comb. We remove the comb to create well for sample loading. Okay, so in the sample DNA, we add sucrose or glycerol, so that it provides some weight and it will go down to the bottom of the well, so that DNA does not come out of the well. Along with that, we add bromophenol blue to monitor the progress of the electrophoresis. And in the DNA, we add ETBR to visualize the DNA. ETBR gets intercalated into the major group of the DNA. Okay, then when electrophoresis will be done, then we will visualize the result of the electrophoresis in UV light. Okay, UV light will excite the electrons of ETBR to higher orbit, and when those electrons return back to their original site, that will emit some light. And this light can be detected. Okay, we can see that light. So in this way, electrophoresis is done. Okay, this is a small topic of gel electrophoresis. There are other types of electrophoresis also. In the upcoming videos, we will see different types of electrophoresis. This is just a part of electrophoresis. It is gel electrophoresis. Other electrophoresis like capillary electrophoresis is there. Okay, so we will see that in upcoming videos. Okay, if you like the video, please share, subscribe, and also hit the like button. Okay, thank you.